So if you're going to have a cell cycle, you'd really better have control over it. To a certain extent, single-celled life forms like yeast or bacteria or some types of protozoa don't really need that much control over their cell cycle. I mean, they got to keep it running properly, but if you're a yeast or a bacteria and you can reproduce, you probably should reproduce. Because, your success is going to be dependent upon how many offspring you can generate. On the other hand, in multicellular life, like, say, us, so we have a term for when cells replicate and go through the cell cycle and go through mitosis out of control and just do it constantly. And that term is cancer. So for any sort of multicellular organism, maintaining control over the cell cycle is essential to maintaining control, to maintaining the continued life of the organism. So the cell cycle has several points of control. And in order for a cell not to become cancerous, you have to really maintain fairly strict control over these points. And there are, technically there are a lot of different control levers. But they mostly come down to three main areas. So let's start here. We're going to start imagining a freshly divided cell, right? So this is a cell, just came out of mitosis, the brand new cell. Right? We know that the first phase is going to be G1 of interphase, which stands for the gap 1 or growth 1, depending upon how you want to look at it. So, as a cell is approaching the end of G1, there is what's called the G1 checkpoint. And this is the checkpoint where the cell is trying to decide, is it supposed to divide or not? Because actually, most of the cells in your body don't divide. Muscle cells, neurons, things like that largely don't divide. You have a pool of dividing cells called stem cells, and when you need replacements, like say it actually doesn't really happen with muscle and nerve cells, but say you need some new skin cells. For the most part, your existing skin cells, most of them don't just divide. What you'll have is a skin stem cell will divide, and then one of the two resulting cells will become a new skin cell, and the other one will stay a stem cell and keep dividing. So most of the cells in your body are actually not supposed to divide. And those cells will go off into a phase called G0, right? If you don't get a signal that says you are a cell that is supposed to divide, then you go into G0, which is kind of like a perpetual state of G1. Right? It's infinite gap. And G0 can be permanent or it can be temporary. Basically, a lot of cells, say your neurons, when they reach this phase or this stop point, they go into G0, they're going to stay there forever. For some cells, it's like they go into G0 and they can stay there indefinitely, but in an emergency situation, they could be reactivated and told to divide. So that would be permanent versus temporary. However, usually it's the rest of your body coordination that is telling cells whether they should divide or not. So the G1 checkpoint usually involves an external signal. Often this is going to be like, say you have a cell, there's the nucleus. This cell will have receptors, and it has a whole bunch of different sorts of receptors on its external surface. And some of these receptors can respond to hormones called growth factors. And if the growth factor binds to the receptor, then that sends a signal that tells you go to S. And if you don't receive the growth factor for long enough, you just sort of go to G0 by default. But the growth factor says to a cell, hey, you are eventually destined to reproduce, so you should prep for that. Remember, DNA is made in S phase. Replicating the DNA is a very energy-intensive process. It takes a lot of energy to do it. And there's no need to do it unless you're going to reproduce. It's like one of those copies is just going to be extra. So if you're not going to reproduce, don't spend the energy to do it. So cells that are not supposed to reproduce, that are not eventually going to be reproducing, shouldn't bother spending all of that energy. But those that do get this chemical signal, this growth factor, will proceed into S and replicate their DNA. So they'll go through S and then get into G2, the second gap phase, and they'll go through G2. The G2 checkpoint usually involves an internal signal rather than an external signal. And this signal is provided by what's called cyclin, which has the word like cyc for cycle right in it, because it is the internal control of the cell cycle. Before you can exit G2 and go into mitosis, you have to pass the G2 or cyclin checkpoint. What this is basically a timer. So cells that, so DNA synthesis is going to take a certain amount of time. And DNA synthesis is not perfect. You make mistakes. All living things make mistakes when they replicate their DNA. And if these mistakes are not fixed, then they result in mutations. Mutations, generally, usually not very helpful. So during G2, one of the main tasks that is performed is repairing those mutations. You have enzymes in the cell that have to go around and hunt out the mutations and then figure out how to fix them. That takes time. So one of the main purposes behind this G2 checkpoint is to make sure that the cell remains in G2 long enough to fix most of the mistakes. It's never going to fix all of the mistakes, but it hopefully will be able to catch most of them. And so that's the G2 checkpoint. And I'll explain a little bit more about cyclin later on. Boom. The third checkpoint is, it says interphase. It should be metaphase here. Metaphase. All right. And remember, in metaphase, your cell has the chromosomes lined up at the midpoint, and it has the spindle apparatus coming out and connecting to each of them. Oh yeah, I call this the metaphase checkpoint. It's technically the metaphase anaphase checkpoint, because it happens at the boundary between metaphase and anaphase. You can't go into anaphase until you pass this checkpoint. So that's metaphase, and here we have anaphase, where you're going to be reeling in these chromosomes. And this is because there's a particular type of mutation called a chromosomal non-disjunction. And a non-disjunction is where you have your cell and the spindle apparatus doesn't hook up properly to one of the chromosomes. And so here, one of the resulting cells will end up with an extra chromosome, a double dose of one of the chromosomes, because it grabbed the whole thing, and one of them will end up missing a chromosome. Both of those cells are going to be seriously screwed up. They're probably going to die, but these are the sorts of mutations that can actually result in cancer, or can result in cancerous cells becoming much more dangerous. So before you can proceed from metaphase to anaphase, there's a checkpoint that checks to see that each spindle line is hooked up properly to the chromosomes. Again, this isn't perfect. Non-disjunctions can still happen. But the fact that there is this checkpoint where you check to, your safety wires to make sure that everything is hooked up properly means that these sorts of non-disjunctions are very, very rare. If the cell doesn't pass the anaphase, metaphase checkpoint, it goes into apoptosis, which is programmed cell death or cell suicide. And this is because, if you've gotten to the point of metaphase and something has so seriously gone wrong that the spindle isn't hooking up properly, this cell should probably not reproduce, right? If it screwed it up once, it might screw it up again. For the most part, none of your cells are that important. Your body is always willing to jettison a single cell if it thinks that that cell might be dangerous. So it's easier to just have this cell kill itself off in an organized way through apoptosis than take the risk that it might become a horribly mutated cancer cell later on. Those are the 3 checkpoints. The G1 checkpoint, usually controlled by an external signal. The G2 checkpoint, controlled by cyclin and internal signals. And the metaphase-anaphase checkpoint, which is controlled by proper attachment of this spindle apparatus. And if you pass the metaphase-anaphase checkpoint, you go ahead and complete the rest of mitosis. There are actually several different types of external signals. Like the easiest one is the growth factor one, like I told you, which controls the G1 checkpoint. But there are lots of other external signals as well. One of them is what's called density-dependent inhibition. With density-dependent inhibition, if you have, like, say, let's say we have an area, and we've got, like, a few cells in it, but they're pretty spread out. They're not touching each other, okay? There's no inhibition here. All right, these cells can grow if they receive the proper growth factor and they pass their cyclin checkpoint. Now let's go away and come back a little bit later. And now this area is like crowded with cells because they've been growing. And you can see how the cells are all running up against each other and touching each other. In this case, what you get is density-dependent inhibition, which is when cells are bumping into each other, that sends a signal that there is no room to grow. Which equals inhibition. Even if these cells are being bathed in growth factor, they will usually stop growing when they bump into each other because there's no more room for them to grow. And so this is density-dependent inhibition. There's also what's called anchorage dependence. Not all cells have anchorage dependence. But most of your cells are like supposed to be in a particular place. Like maybe you've got a whole bunch of liver cells in your liver. They're supposed to be in your liver. And the cells in your liver don't just like float around in your liver in a big mess. They're attached to what's called the extracellular matrix, right? So the extracellular matrix is this network of protein fibers that the cells are embedded in. And so the cells will be bound to each other, and they'll be also touching this extracellular matrix. And they have to receive the signal that they are bound to the extracellular matrix, in order to divide, right? They have to be embedded within this matrix, and that's because, like, your liver cells are only supposed to reproduce in the liver. What happens if one of these cells falls off? and just gets by itself. Maybe it falls into the blood and it gets carried over to the kidney. Well, do you ever want to grow kidney cells in your liver cells in your kidney? No. There is no circumstance under which you should be growing liver cells in your kidney. So cells that are largely supposed to remain stationary, which is most of the cells in your body, can only reproduce if they are anchored to the right place, wherever it is that they are supposed to stay. And these two things are both to prevent the growth of cancer. And in order for cells to become cancerous, they have to lose both of these dependencies. Right? Cancer, by definition, will continue growing even if there's no room to grow. That's why cancer grows into a lump. Like, that's what happens when you have a big clump of cells that keeps growing despite the fact that there's no room for it to grow. It just pushes everything outwards. So cancer cells have to have lost density-dependent inhibition. They don't stop growing when they run into other cells. Now, tumors, which are like cancers usually start off as tumors, right? Tumors are cellular growths that grow without control. But in order to be considered a true cancer, you have to be malignant. And malignancy means that the cancer can spread. And one of the ways that cancer spreads is like a little cell falls off of the cancer and goes somewhere else in the body, and when it lodges, it starts to grow there. So the anchorage dependence prevents cancer from spreading because, like, the cancer can only grow, or the cells can only grow, when they are anchored in the right place. If they fall off and go somewhere else, they can't grow anymore. So in order for a cancer to become malignant and to spread, it has to lose anchorage dependence, right? And both of these are situations where, like, a protein which is responsible for this must be mutated. So the reason why cyclin is called cyclin is because it happens in cycles. So if you take a look at this picture up here at the top, right, we'll see several cell cycles. Here you can see this is a mitosis. Then we go to G1S, G2, mitosis, G1S, G2, mitosis. So we got three cell divisions taking place here. The cyclin is this purple line, right? As soon as you finish mitosis, you not only stop making cyclin, but you get rid of, you destroy all of the cyclin that you have. So cyclin goes back, to 0. Once you start, or at least get to the middle-ish of S phase, one of the things that happens is you start producing cyclin. And you just generate it at a slow rate, but it doesn't go away. So you start building it up and building it up and building it up. This only happens in S phase or later. So cells that go into G0, cells that are still in G1, they don't do this. You only start making cyclin once you have passed the G1 checkpoint. And cyclin builds up, builds up, builds up, builds up, builds up. And when cyclin reaches a high enough concentration, it will allow you to go past the G2 checkpoint and into mitosis. Well, sort of. Actually, what you have is what's called MPFs. And when cyclin reaches a high enough level, you start making these MPFs, and they are what kicks the cell into mitosis and gets it through mitosis. And at the end of mitosis, boom, both of these things reset to zero. So that's why cyclin is a clock, right? However long it takes for cyclin to build up high enough to start activating the MPFs, that's how long you spend in G2, or the later half of S phase. Here you can see the cycle going in a circle, right? So if we start here at the end of mitosis, there is no cyclin, right? You have what's called CDK, which is a thing that binds to cyclin, and it's the other half that makes MPF. So you have the CDK. You go through G1. When you pass the G1 checkpoint, you begin to accumulate cyclin. You get more and more and more of it. The cyclin binds to the GDK or CDK, and those two things together make MPF. And when you've got enough of this MPF, you pass the G2 checkpoint into mitosis, and then in mitosis, you degrade the cyclin, and the process starts over. So it's like rest, build up, 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 mitosis, rest. So, just to reiterate, in order to control the cell cycle, we have chemical clues or cues, CDKs, cyclins, also external chemical cues like growth factors, and we have physical cues like density-dependent inhibition and anchorage dependence. All of these exist to make sure that cells don't reproduce unless they are supposed to reproduce. When you have cells that reproduce when they're not supposed to, what you get is uncontrolled cell division, which leads to tumors and eventually cancer.